In this video, we will explore protein targeting and sorting, essential for cellular function. We'll study chaperones, tools to study these processes, and how defects impact diseases. Specialized cells and implications will also be discussed. This comprehensive script aims to provide a detailed understanding of protein sorting mechanisms. Let's start. Protein targeting within cells is a complex process that requires chaperones, specialized proteins that assist in protein folding and delivery. Chaperones recognize specific regions on newly synthesized proteins and prevent their aggregation. They also aid in translocation across membranes and delivery to organelles. Dysfunction of chaperones leads to protein misfolding diseases, emphasizing their importance in maintaining cellular protein homeostasis. Post-translational modifications significantly influence protein sorting within cells. One such modification is protein glycosylation, which involves the attachment of carbohydrate moieties to specific amino acid residues. Glycosylation can occur in the endoplasmic reticulum and Golgi apparatus and has a profound impact on protein stability, folding, and targeting. Owing to the diverse array of sugar moieties and glycosylation patterns, this modification serves as a code that directs proteins to their appropriate subcellular locations. Various lectins and glycosyltransferases participate in this recognition and sorting process. Notably, aberrant protein glycosylation is implicated in numerous diseases, including cancer and neurodegenerative disorders, underscoring the importance of glycosylation in protein sorting. Nuclear proteins perform essential roles in cellular processes such as gene expression, DNA replication, and repair. However, they must first traverse the nuclear envelope to gain access to this compartment. Nuclear import and export processes are highly regulated and mediated by specific transport receptors, such as importins and exportins, respectively. These receptors recognize nuclear localization signals, NLSs, or nuclear export signals, NESs, present on the cargo proteins. Nuclear pore complexes, NPCs, large proteinaceous structures spanning the nuclear envelope, serve as gateways for nucleocytoplasmic transport. The intricate interplay between transport receptors, NLSs/NESs, and NPCs ensures precise and selective nuclear protein import and export. The Capille vesicular transport system controls protein movement between the endoplasmic reticulum, ER, and Golgi apparatus. Cargo proteins for the Golgi are sorted using Capille coat components. The SAR1 GT-PASE recruits coat proteins Sec23/24 and Sec13/31, which form a cage-like structure around cargo, allowing them to be packaged into vesicles. Cargo proteins directly interact with coat components to ensure correct targeting to the Golgi. In the secretory pathway, proteins are usually moved forward from the ER to the Golgi and beyond. However, sometimes proteins need to be sent back to earlier compartments. Retrograde protein trafficking retrieves proteins from the Golgi to the ER or the ergic. This process involves specific signals on the proteins, like D-lysine or D-arginine motifs. Retrograde transport relies on coat complexes like COPI, which form on the Golgi membrane and help create retrograde transport vesicles. This mechanism ensures the proper recycling and maintenance of proteins in the secretory pathway. The ubiquitin proteasome system is essential for removing damaged or misfolded proteins. Ubiquitin is attached to target proteins, marking them for recognition and degradation by the proteasome. This process is crucial for regulating protein levels and facilitating cellular responses. Autophagy is a cellular process that breaks down and recycles cellular components. It involves the formation of double membrane structures called autophagosomes, which encapsulate cytoplasmic cargo. These autophagosomes then fuse with lysosomes, leading to degradation. ATG proteins play a crucial role in this process. Dysregulation of autophagy is associated with diseases like neurodegenerative disorders and cancer. In specialized cells, such as neurons and immune cells, protein sorting and secretion mechanisms play a vital role in their proper functioning. Neurons use specific machinery and motor proteins to sort and transport synaptic vesicles for efficient synaptic transmission. Immune cells store, sort, and release signaling molecules through maturation and polarization of secretory granules. Precise understanding of these processes is key in unraveling cell function and dysfunction. Disruptions in protein sorting mechanisms can lead to severe consequences and are associated with various diseases. For example, mutations in genes encoding chaperones or trafficking machinery components can result in protein misfolding diseases, such as Alzheimer's disease or cystic fibrosis. Defects in protein trafficking have also been linked to autoimmune diseases, cancer metastasis, and neurodegenerative disorders. Furthermore, impaired protein degradation pathways can contribute to the accumulation of toxic proteins in neurodegenerative diseases like Parkinson's or Huntington's. Understanding the underlying causes and mechanisms of protein sorting defects provides valuable insights into the pathogenesis of these diseases and potentially identifies therapeutic targets for intervention. The endolysosomal system comprises a network of interconnected compartments involved in protein sorting, degradation, and recycling. These compartments include early endosomes, late endosomes, and lysosomes. The routing of proteins through this system relies on a complex interplay of vesicular transport, fusion, and fission events. Rab-GT possase, tethering factors, and snare proteins coordinate membrane trafficking and fusion, ensuring the correct delivery of cargo to their respective destinations. In addition to protein sorting, the endolysosomal system also plays a crucial role in maintaining cellular homeostasis by regulating nutrient uptake, membrane turnover, and organelle quality control mechanisms. Protein sorting is crucial for cell polarity and tissue development. Signaling pathways help distribute proteins needed for polarity establishment, like receptors and transporters. Small GT possase, CDC42 and PAR3, control protein trafficking and localization. Proper protein sorting is vital for tissue organization and functionality. Defects in sorting can cause developmental issues and disease. Recent advancements in imaging, proteomics, and molecular biology have revolutionized the study of protein targeting and sorting. Techniques like super-resolution microscopy, proteomics approaches, and genome editing technologies allow researchers to visualize cellular structures, analyze protein interactions, and manipulate genes to understand the complex mechanisms of protein sorting in cellular function and disease. Protein targeting and sorting are vital processes that regulate protein localization and function in cells. Chaperones, modifications, vesicular trafficking, and degradation mechanisms determine efficient protein sorting. Dysfunction leads to diseases and impaired cellular function. Ongoing research and emerging techniques enhance our understanding and provide therapeutic opportunities.